For this discussion, we're going to look at several examples of how major changes, major innovations have been made with only little tiny changes in the DNA. And I want to bring up this idea that was posed a long time ago. This is post-Darwin, but was posed and then later kind of rejected and laughed at. But then it's been revised as we've understood how evolution and development works. And this is the idea of the hopeful monster. So I'm actually going to add that here because some of these maybe fall into this category. So one of the, it's kind of a misconception, but one of the criticisms of Darwin's work was that changes are just going to result in bad things, which is actually true most of the time. And Darwin agreed with that and said then most of those bad things would then be removed and we would never see them long term. But people argued and said, oh, you know, we've got these mutations, these terrible, awful things. And it's a little bit of a misunderstanding of what a mutation is and how they work. But they can be bad. And so the idea is that occasionally a single change would result in a dramatic phenotypic shift, a mutant basically that looked very, very different from anything else in the species. And the hopeful monster idea is that every once in a while this massive change might actually result in a benefit in something good. So we're going to look at the Repression of Dll by UBX. And it's a little bit of a simplification because abdominal A plays a major role here also. But I'm just going to remind you of this kind of dichotomy. So here we go. This is a fruit fly. And UBX is in red. And notice that right there where we have that strong UBX expression, that represents the boundary between the thorax and the abdomen has no limbs at all. And so UBX represses the limbs growing in the abdomen. That's a unique, distinct thing in insects. If we look at the ancestor of all arthropods, we do have UBX only a little bit in the most posterior region, so not really important in the rest of the body. This is a crustacean. And so in this crustacean, we can see that UBX is expressed all the way in the body, but it doesn't repress distillus. So we basically have a shift from an ancestral form of just very posterior distillus expression to shifting all the way forward. Now remember that shift, and I'm not even sure this has been traced yet in these early crustaceans, but that shift could either be from a regulatory element in the UBX itself, or perhaps from something upstream that caused that dramatic shift. But that's a big change, okay? But in the long run, notice it doesn't have much of a difference on body form, right? The body form of this and the early crustaceans is a head and a homonymous trunk. Here we have a head and a homonymous trunk. So this first initial shift in range did not have a dramatic impact in the overall body plan of arthropods. However, later UBX then takes on additional functions and additional roles, and that's when we start to see this shift. And so to do this, scientists began to look at this very posterior part of the UBX gene. And this is a little bit of an exception to the rule, because the rule is most of the time changes in these sorts of things are going to be a change in regulation and not necessarily a change in the protein coding itself. However, in this case, where scientists very early on began to focus on the protein. And so these are amino acid sequences at the terminal end, right, the C terminus, that's where the end of translation occurs. And they started focusing in on this. And what they noticed was when they looked at all of the insects, they had this shared kind of motif where they have the same similar, the QA motif, and then the K followed by lots and lots of A's. And so in all those organisms where distillus is repressed by UBX, UBX has this really distinct feature. And that kind of jumps out, right? Even if we don't have any sort of direct evidence that this represses distillus, that this specific sequence is what gives UBX the ability to repress distillus, it's still kind of like a smoking gun, something that you would want to investigate. And so if you look at the Ron-Shagan paper, which we read in an earlier discussion, they actually go in and they do some manipulation of UBX, and then they look at it in Drosophila. They make some hybrid UBX ones that are part shrimp, part fruit fly. They make single changes, and they're able to trace this to just a few residues right here. Okay, but notice that in the crustacean, the myriapod, the chelicerates, so phylogeny coupled with the genetic data from those species gives us a roadmap, a beginning to figure out what it is. And in this case, it's one of those kind of minority rules where a change to UBX itself is what gave it the ability to repress Dll. Okay. Another thing which we're just beginning to understand is tricky because we don't have any living modern species that have the ancestral form, but it's thought from the fossil record that the ancestral form of wings in insects was basically these lobes that were across the entire body and then reduction to just the thorax. But notice in this primitive one that we have 1, 2, 3 sets of wings and then little remnants going back. And there's even some vestigial remnants of these anterior wings, but fairly early on in insect development, we lose the anterior wings, the ones that are in the first thoracic segment, and we end up with just 2 pairs of wings. Then later there are lots and lots of models. And so insect wing number has gone through multiple variations. We don't know all of the genetic details, particularly these ancestral ones, but we're beginning to understand wing modification and diversity. So let's look at some of the diversity. So first of all, I want you to understand the ground plan. The ground plan for flying insects is 2 pairs of wings. Now again, there might have been some remnant, but there's arguments as to whether they were ever truly fully powered in flying wings when they had 3 pairs, but certainly there are 2 pairs in the earliest really good flyers and from most of the fossil records we have. And many insects maintain this ancestral form. So dragonflies, for instance, represented by these 2 separate species, have this ancestral form. And for a long time, all insects had 4 wings. Later, there were different modifications. So dragonflies, and probably the earliest flying insects, actually use their wings separately. So when the front wings beat down, the hind wings are beating up. And so we have this kind of alternating wing beat pattern. However, in many other insects, that's changed. Either the wings are linked together like in butterflies so that they go up and down together. Most of the more complex holometabolous insects have those linked wings when they fly. Or 1 pair of wings has been highly, highly modified, and the Hox genes play a key role in this. So for instance, the 4 wings of beetles are modified into these protective shells, and the hind wings look much more like the ancestral form. They're membranous and flap up and down and have powered flight. In the butterfly, we have these dramatic modifications and color patterns that form in both the forewing and the hind wing. And so people began to use this as a natural laboratory for the study of evolution and development. Here is a robber fly, right? And Diptera, this is just one of many Diptera, fruit fly or another Diptera. They've got this unique form. I picked this picture, 1, because robber flies are cool. This one's caught a spider and is having a snack. But number 2, you can see the 4 wings, which are here in profile. This long black line represents these 4 wings, which are for powered flight in the Diptera. And then the hind wings, which have been modified into these sensory organs, these halteres. And the haltere forms or functions as a little gyroscope. It spins around in circles when they fly, and that way when their orientation changes, it takes a little while for the spinning halteres to catch up, their momentum to catch up, because the spinning maintains their old momentum. And the difference between the body's momentum and the haltere's momentum allow the fly to interpret position and do kind of all these great acrobatics that we see flies do, like landing on ceilings or avoiding your hand when you try to swat them. And so, of course, when we modify UBX, one of these Hox genes, and we turn off its expression in the thorax, we end up with a more ancestral form. And so this was really cool. This is really the hopeful monster, right? All we had to do was change one small thing, change the UBX expression and change its target. Maybe that's 2 things, but where and the target, where UBX is expressed and the target of UBX, and we get this dramatic shift from 4 wings to 2 wings. Okay? And so we've got 2 wings to 4 wings to 2 wings in the fly. We've got dramatic modification of butterfly wings, and we'll look at that in a later chapter about how that comes about and the steps along the way there. And then we have the beetle, which is another example. Now the cool thing is UBX is involved in all of these. So UBX is expressed only in the hind wings. In the dragonfly, even though it's expressed there, it doesn't play any role in differentiating the hind wings. So it doesn't really have a wing function in the dragonfly. But it's gained a wing function in the beetles, where it maintains ancestral diversity, and a separate one in the fruit fly and other diptera, where it modifies those hind wings into halteres. So great example of a single gene with multiple outcomes and multiple different effects. So this really brings home that idea of a limited tool set, right? Single gene, but gaining new abilities to target new genes, or changing the protein pattern, the protein sequence itself, so that it can do new things. So just review and make sure you know the UBX example in insects. Okay, same gene, different morphologies, and again the difference is because it's targeting different downstream genes. So in the ancestral gene, UBX only targets a number of genes, and they don't really do anything that are specific or at least drastic for those hind wings. They still look pretty much like the forewings. But in diptera, we've gained these extra abilities that modify into a haltere. In butterflies, we've gained these extra abilities that begin to recruit genes that grow the scales on butterfly wings and recruit pigments for spots and stripes and all these beautiful colors that you see in the butterfly wing. So you should be able to explain how a single gene can result in different morphologies. It's fairly simple, right? We just have different target regions that are available where, and it's still expressed in the same area, but it's targeting and turning on different genes in these different taxa. Okay? Example number 4. Many larval insects have legs not only in the thorax, right? So these are thoracic legs, but they also have these abdominal legs. Now remember, I told you the basic insect body is that we don't have appendages in the abdomen, and this holds true. There are some exceptions to this. And the butterfly larvae are one. As adults, they look like a standard insect. But as the larvae, they've got this unique form where their abdominal segments have legs, essentially, appendages. Now notice they look quite different, and they actually are morphologically quite distinct and different from the thoracic legs. So the question was, if we have this model of Distillus being expressed in the abdomen, and then UBX being expressed in the abdomen, and then UBX targets Distillus to repress it, what's going on in the butterfly larva? Has UBX modified its ability to repress Distillus so it can't do it in the butterfly? That didn't really make much sense because we still have segments, abdominal segments 1 and 2 and 7 and 8 that still aren't doing it. So something is still repressing Distillus, but we've kind of relaxed this repressive effect in segments 3 through 6. So people started studying this, started looking at it, and and the breakthrough was when they looked at butterfly larvae in really, really close-up view. Okay? And so here we have it. So they said, hey, maybe it's not Distillus, maybe there's some other gene that is growing these different looking appendages. So the first thing they did was they stained for Distillus. And sure enough, Distillus, here's in this dark kind of purplish color, Distillus expressed in the mouth parts, in the thoracic legs. It's also expressed in the abdominal legs. So Distillus is still coming on and doing its ancestral function of growing appendages. But for some reason, UBX isn't repressing Distillus, even though it seems to be expressed in this area. And so then they said, let's take a much closer look at the expression of this UBX gene. And what they found were these little tiny regions. I think I got it zoomed in, or maybe I don't. But these little tiny regions, notice these almost like bullseye areas, where Distillus is not expressed. And so we have this tiny little shutting off, where UBX is not expressed. This is a stain for UBX. So UBX is not expressed in those few cells. That allows Distillus to initiate, come on, and begin appendage growth. So rather than changing UBX's connection to Distillus, we simply repress UBX in the correct cells where the appendage needs to grow. And again, a random mutation that was a benefit for the butterflies and has been kept and become a standard feature of butterfly larvae. Now, one last final example. Let's look at some vertebrate examples. And although it's much, much harder to do in vertebrates because embryos are more difficult to work with, they're more ethical considerations, and so really we still use insects as kind of a model, a laboratory for study of development. So you'll probably see a question on the exam like that, like what group is the best kind of living laboratory model for looking at how developmental genes change morphology and how we get evolution of all these diverse things. It's really in arthropods. And we're beginning to gather a little bit of data in vertebrates, but it's much harder. Like in humans, right? They really ethically can't do these sorts of experiments in humans, human embryos. But we could do them in chickens, and there has been some work done in chickens. But one of the cool things is, at least in general outlines, it's the same story. So ancestrally in the tetrapod arms, we have a very simple control mechanism and then not much diversification. The forelimbs look just the same as the hind limbs. But in different lineages, we have the same gene expressed in the same area, but now being able to target more downstream genes. And so we can get a more complex series of cause and effect occurring that are going to diversify and change the morphology. And this gene and different gene targets explain the differences, not only the initial complexity of these organisms, but also the differences between these vertebrate forelimbs. Okay, so quick review. Number one, major shifts in expression domains result in major differences. So the snake is kind of a hopeful monster. We had this one shift in where HOXC6 was expressed, and boom, you have an organism with its thorax the whole way. Now, maybe you would argue it's not a single step, because we'd have to have that step and then later the reduction and loss of legs. And so, yeah, it's really maybe multiple steps, but still not like tiny little steps. It might be 2 or 3 major shifts that result in this incredibly successful and diverse group of organisms. Those, in addition to having shifts in domain, these developmental genes can also gain new targeting abilities, and that increases complexity. And then finally, sometimes little minor shifts that we may need to look at very, very closely might result in a major phenotypic shift. So little genetic changes, little changes in expression domains can have a major impact downstream in the way that organisms look. So review those examples, make sure you know them, prepare for them. You'll see something about them on exam number 2, which is coming up on Tuesday. I believe it's the 31st of March through the 2nd of April.